Assalamu alaikum, I'm Marwa. We'll talk about female reproductive physiology. This is our first lecture in this semester. Lecture content divided into three sections. Today we'll discuss ovarian follicle development, followed by physiology of puberty and then menstrual cycle. Lecture objectives will define folliculogenesis and its growth from primordial follicle to graphian follicle. Detail morphological change in this developmental stage will explain regulatory mechanism for folliculogenesis and pathology or abnormalities in its form of PCOS, which stands for polycystic ovarian syndrome, and premature ovarian insufficiency. By the end of the lecture, we'll define and explain these terminologies. First of all, we'll talk about primordial germ cells, from which it arises from epiblast of bilaminar embryonic disc. What is a bilaminar embryonic disc? If we divide early intrauterine development, it divided into three stages: germinal stage, embryonic, and fetal stage. Germinal stage, which is the first 2 weeks of intrauterine development, it's involved from fertilization up to implantation and first 2 weeks, followed by embryonic stage, which is from 3rd to 8th week, which is for organogenesis. And the last stage is the fetal stage from 9 weeks up to the term, which is the 4th weeks. Now we talk about the germinal stage, first 2 weeks. At that stage, the fetus consists of 2 layers, bilaminar disc, 2 laminae or 2 layers, epiblast and hypoblast. We'll talk about this stage. Bilaminar disc embryo. This is epiblast and hypoblast. Epiblast, this is cells, is epiblast. From this arise al primordial germ cells. Tamam? Okay, primordial germ cells arise from epiblast and move to the yolk sac, wall of the yolk sac, by 4th week. It's developed or arise from the 2nd week. At the 4th week of gestation, it moved to yolk sac. Then migrate to the final destination, the hindgut, to reach genital ridge by 6 weeks. All of these occur by the end of the 6 weeks. It reached the genital ridge. Okay, primordial germ cells arise at the 2nd weeks. By 4th weeks, it migrates to yolk sac. Yolk sac is early structure for nourishment before placenta is formed. And then from yolk sac, it migrates to hindgut at this side. And from the hindgut to its final destination, genital ridge. Here. So, origin from epiblast at 2nd week, migrate by 4th week to the yolk sac, and final destination genital ridge by 6 weeks. Okay, genital ridge, it's a future gonads. The gonad develop into ovary in the female in the absence of SRY gene. SRY gene stands for sex determining region in Y chromosome. Y chromosome in male. It's okay. If this Y chromosome is absent, and so on, the SRY gene is absent, the gonad will develop into ovary, and the germ cell develop into oogonia. On the other hand, in the male, these gonad develop in testes, and germ cells become spermatogonia. The primordial germ cells undergo rapid mitotic division for replication to increase their number and form oogonia up to the 5th month of gestation to ensure sufficient stock for gametogenesis. Gametogenesis is the developmental process to form mature ovum from these oogonia. The majority of oogonia differentiate into primary oocytes, all of these intrauterine. It's okay? Before birth. So, primordial germ cells develop into oogonia, and oogonia differentiated into primary oocytes. In the primitive or fetal ovary, granular cells surround these primary oocytes to form primordial follicles. We have to differentiate between primary oocyte and primordial follicle. To understand the difference between follicle and oocyte, we start from primordial germ cells. These primordial germ cells underwent mitosis to replicate and increase in number. It's okay? After mitotic division, it's form oogonia. Majority of this oogonia will die an atresia or degenerative death, and the rest of oogonia will enter meiosis 1. When oogonia enter meiosis 1, it's form what we call primary oocyte. This primary oocyte, this is pink cell, when surrounded by granulosa cell, these blue cells, when it's surround primary oocyte, this is called primordial follicle. So primordial follicle is involve both primary oocyte and granulosa cell. All of these states is intrauterine up to primordial follicle. As we say, primordial follicle, it's replicated by mitosis up to for 20 weeks of gestational age to increase their number. So at that gestation, it's a maximum number 6 to 7 million of primordial follicles. From 6 to 7 million at 5th month, at birth it's only 1 to 2 million. These atresia continue throughout life, and by menarche at puberty only 300 to 400,000 are present, and at menopause, which is around 50 years of age, is none. To differentiate between oocyte and follicle, we revise this diagram. Oocyte is a cell which is developed into ovum through meiosis. The development process of oocyte is called oogenesis. Oogenesis involves oogonia, which is differentiated into primary, then secondary oocyte, otid, and the mature ovum, which is ready for fertilization. Ovum has haploid nucleus, cytoplasm, vitelline membrane, protective space, zona pellucida, and corona radiata. Ovum contain female nucleus, which is fused with sperm and fertilized into zygote. Follicle secretory cavity surrounds the developing oocyte. Developmental process of these follicular cells called folliculogenesis. This folliculogenesis involves primordial follicle developed into primary, secondary, then tertiary follicle, and lastly Graafian or preovulatory follicle. Follicle consists of oocytes and surrounding cells. These surrounding cells consist of granulosa cells, theca cell, antrum cavity filled with follicular fluid. Following ovulation, the follicle remain in the ovary and develop into corpus luteum. And so folliculogenesis and oogenesis are parallel independent processes occur in the ovary from fetal life and up to menopause throughout the reproductive age. As we say, oogenesis is a maturation of female gamete starting at fetal life, pause, and then resume at puberty, produce ovum ready for fertilization, while folliculogenesis is maturation and development of the follicle surrounding oocyte from primordial to dominant preovulatory Graafian follicle. Function of these follicles provide nutrient and hormonal support to the developing oocyte. In this slide, folliculogenesis explained in stages. First, start with primordial follicle, which is the dormant or resting stage. Contain oocyte with single layer of granulosa cells. Granulosa cells in this stage, it's flat, squamous, single layer. When these granular cell layers become cuboidal, it's called primary follicle. And when surrounded by multiple layers of granulosa cells and theca cells layer start to develop, it's called secondary follicle. As shown in this picture, Primordial cell with single flat layer of granular cells. Primary follicle, these granular cells become cuboidal. From squamous, it's cuboidal now. But it's a single layer still, تمام? In secondary follicle, it turns in multiple layers of granular cell, and this yellow layer, it's theca cells. So, in secondary follicle, it's oocyte surrounded by multiple layers of granular cells and theca cells. Theca cell is vascular layer, provide nutrient for growing follicle and oocytes. On the other hand, granular cell layer, it's like these capillaries. After primary and secondary, it's tertiary or called antral follicle. Tertiary follicle differ from secondary in that tertiary follicle have a cavity, fluid-filled cavity called antrum. When the follicle develops this antral cavity, it depends on its growth on gonadotropin. Gonadotropin stands for follicle stimulating hormone and luteinizing hormone, which produce from anterior pituitary gland. So, tertiary follicle develop receptors for FSH and LH for further growth. Last stage of folliculogenesis is dominant or pre-ovulatory follicle, with the follicle dominate over the rest of antral follicles. If we suppose in each menstrual cycle, group of antral follicles respond to FSH, follicle stimulating hormone, and start to growth, one of them will dominate over the rest of antral follicles and become dominant pre-ovulatory follicle up to 20 mm in size. And this follicle will ovulate at the mid-cycle in response to luteinizing hormone. The rest of antral follicles, which is not dominate and will not grow to pre-ovulatory follicle size, will die through atresia. In this picture, we demonstrate primordial, primary, secondary follicles. And this antral follicle or tertiary follicle contain antral cavity with follicular fluid. And from this stage, it responds to FSH. Okay? According to gonadotropin, we can divide folliculogenesis in two phases, gonadotropin-dependent and independent phase. Pre-antral, which is contain primordial, primary, and secondary follicle, is gonadotropin-independent phase. This stage of growth depends on growth factors produced locally by the follicle. And from antral or tertiary follicle, the growth depends on follicle stimulating hormone, which is released from anterior pituitary gland. By now, we discussed folliculogenesis in detail, stages and phases. So, we'll talk about gametogenesis. Gametogenesis is a broad term, contains oogenesis and spermatogenesis according to the gender. In female, it's called oogenesis, and in male, called spermatogenesis. Gametogenesis is a process of maturation of oocytes, which is undergo cytoplasmic chromosomal change to form definitive ovum. The end result of production is haploid gamete, which contains 23 chromosomes from diploid germ cells through meiosis and differentiation. Oogenesis differs from spermatogenesis in that oogenesis is a cyclical event, almost monthly, from puberty up to menopause in female, while spermatogenesis begins at puberty and continues throughout the adult life. Germ cells, through mitosis division, produce genetically identical diploid cells to amplify their population before meiosis, to increase their number. تمام? But will be identical and diploid, contain 46 chromosomes, or 2N. While meiosis involves two divisions, produce 4 genetically diverse haploid gametes for sexual reproduction, which is not identical. This is the other difference between oogenesis and spermatogenesis. The end result of meiosis in female oogenesis, it's one mature ovum. Meiosis division allow random assortment of maternal and paternal chromosomes and redistribution of genetic information through process of crossing over. And so each gamete produced will be distinct and not identical in each other. Oogenesis begins during fetal period, but it's not completed until after puberty, as we know. The majority of oogonia proliferate by mitosis and differentiate into primary oocytes. Before birth, part of this oogonia will die by apoptosis, and the remaining of oogonia will differentiate into primary oocytes. This is before birth. These primary oocytes will form primary follicle when surrounded by granulosa cells. At this stage, granulosa cell layer will be flat, single layer of granulosa cell. All of these will be before birth. At birth, most of surviving primary oocytes enter prophase of first meiotic division. Oogenesis contains two meiosis divisions. The first meiotic division will be started before birth and arrested at prophase 1 until puberty. This inhibition will be maintained by peptide hormone called oocyte maturation inhibitor. It's a small peptide produced by follicular cells from birth up to puberty. At puberty, meiosis 1 will be completed, produce secondary oocyte and first polar body. This diagram demonstrates oogenesis and folliculogenesis. Before birth, primordial germ cells differentiated into oogonia. And this oogonia will replicate by mitosis to increase their number, and mitosis will produce identical diploid cells, 2N, contain 46 chromosomes. Before birth, this oogonia will differentiate into primary oocyte. Primary oocytes will enter first meiotic division and arrested in prophase 1 from birth up to puberty. At childhood, ovary is inactive. تمام. At puberty, these primary oocytes will continue and complete first meiotic division to produce secondary oocyte and first polar body. The end result of first meiotic division is two asymmetrical cells. The first called secondary oocyte, which is a large cell, and the other cell called first polar body, which is a smaller cell. This asymmetry due to unequal division of cytoplasm. Secondary oocyte retains the most of cytoplasm and organelle, while first polar body contain little. In spite of that, both cells contain same chromosomal number, will be haploid, contain 23 pairs of chromosomes, but asymmetrical in size. First polar body will die by apoptosis, while secondary oocyte will enter second meiotic division, meiosis 2. Secondary oocyte will not complete this second meiotic division and will be arrested in metaphase 2. At this stage, will ovulated in response to luteinizing hormone. This second meiotic division will not complete it unless this oocyte fertilized by sperm. As we see in the last part of the diagram, secondary oocyte following second meiotic division will result in ovum and second polar body. This second polar body will be smaller, will die by apoptosis also, and mature ovum will be fertilized by sperm to produce zygote. The zygote will retain the chromosomal number, will contain 46 pairs of chromosomes or 2N, while secondary oocyte and mature ovum will be haploid in number. This is the repetition. At puberty, primary oocyte will complete first meiotic division to produce secondary oocyte and first polar body. Secondary oocyte start their second meiotic division and arrested in metaphase 2 up to ovulation. Second meiotic division completed all if secondary oocyte fertilized by sperm. The outcome of second meiotic division is a mature ovum and second polar bodies. These polar bodies will degenerate through apoptosis. In a summary, stage of gametogenesis first start with multiplication phase, in which primordial germ cells underwent mitosis to increase their number. and differentiate into oogonia. Followed by growth phase, this oogonia increase in size, accumulate nutrient to become primary oocytes, which is diploid in number. This is intrauterine and before birth. Even will start their first meiotic division but will be arrested in prophase 1. This is the first meiotic division will complete at puberty, producing secondary oocyte and first polar body, both of which haploid. But secondary oocyte retain large amount of cytoplasm and will continue to enter second meiotic division, while first polar body will degenerate by autosis. Second meiotic division will result in ovum, mature ovum, and second polar body, both of which will be haploid. Second polar body will degenerate and ovum will be ready for fertilization. In this diagram, we discuss oogenesis, and now we revise follicle development or folliculogenesis. Folliculogenesis in fetal life consists of primordial follicle, which is contain oocytes surrounded by single flat layer of granulosa cells. This single flat layer of granulosa cell will develop into single cuboidal layer in primary follicle, followed by multiple layers in secondary follicle and surrounded by theca cell layer, which is the vascular layer. At tertiary or antral follicle will develop antral cavity filled with follicular fluid. This antral follicle depends on FSH on its growth to produce dominant pre-ovulatory follicle, one dominant and the rest will be atretic. This pre-ovulatory follicle or dominant follicle will respond to luteinizing hormone and ovulated at the mid of menstrual cycle. At ovulation, secondary oocytes will be released and the remaining granulosa and theca cell will remain in the ovary to form what we call corpus luteum. At the end of this lecture, these are examples of pathology or abnormality of folliculogenesis. Two examples: polycystic ovary syndrome and premature ovarian insufficiency. In polycystic ovary syndrome, there is arrest of antral follicle growth and failure to form dominant or pre-ovulatory follicle. As we say, each menstrual cycle, group of antral follicle will respond to follicular stimulating hormone and start to grow and develop into dominant pre-ovulatory follicle, right? But in PCOS, the antral follicle failed to develop into one single dominant follicle to ovulate, will be arrested at that stage, will be arrested at antral follicle stage, each menstrual cycle. So the ovary will be cystic in appearance and subsequent subfertility. In premature ovarian insufficiency, there is accelerated exertion of follicle pool, either by excessive activation of primordial follicle growth or by excessive apoptosis of these follicles. And so the follicle pool, which is 300,000 to 400,000 at puberty, will be exhausted, completed before the suggested age of menopause. By definition, before 40 years, the patient will be menopaused. These abnormalities of folliculogenesis. Thank you.